Thanks everybody for coming out. Thanks for having me. It's really been a great day of conversations and interesting things to talk about. Some scientific surprises, which are great. I felt very welcomed. I appreciate it. It's my first time in England, actually. I'm not sure what kept me away. Maybe it was the language barrier. It's nice to be here. And I'm going to tell you today about some research done on fluid dynamics of brain clearance at the University of Rochester. This work is highly collaborative, as you will see. And so I want to start by acknowledging and expressing my gratitude to my senior collaborators. So Jack Thomas sits in mechanical engineering with me at U of R and has a whole career of fluid dynamics expertise, and his ideas are all over this work. And Mike Edergaard sits in neuroscience at U of R and is the person who came and found us and plugged us into these very interesting problems about 6 years ago. And it's also her team that produces all of the in vivo measurements you'll see today. So we engineers do modeling and analysis and this sort of stuff, and they do the in vivo work. Okay, so we're thinking about fluid dynamics of brain clearance. And the first big idea that I want to introduce to you, which maybe you've thought about already, is in the brain, there are two distinct fluid transport systems. There's blood circulation, and there's also a system that circulates water-like fluids. Sometimes it's called cerebrospinal fluid, sometimes it's called interstitial fluid, circulates water-like fluids around and through brain tissue, and has important physiological and biological implications. That's what we're studying. And you can see that in action lots of different ways. Here's one. You're looking at the middle cerebral artery of a live mouse atop the brain. You can see red blood cells rushing along in the artery. You can also see the surrounding cerebrospinal fluid, which we've dyed here. It appears blue. And you can tell that it's moving because we've injected 1 micron fluorescent tracer particles, which are then swept along as the flow proceeds. And the engineer's big contribution here is to use our automated particle tracking code to find all the bright dots and connect them throughout space and time. So we can draw on these colorful path lines, which were not, of course, part of the original in vivo imaging. But okay, these water-like fluids are flowing along, and you can see it if you look at the spaces surrounding arteries in the veins and the brain. Another way to see it is to zoom out and look at a whole animal's brain and inject some tracer dye, say in the cisterna magna in the back, because it's a large cavity, it's easy to hit. And if you do that, what you'll observe is that the tracer spreads over time from the cisterna magna around first the base of the brain and then up around the edges to the dorsal bits, and then into brain tissue. It spreads much faster than diffusion alone could do. It spreads along particular pathways, which look very much like the arterial network, not because this tracer is in the arteries, rather because it's in these perivascular spaces that surround arteries, like the one you were just looking at. That's also in a mouse. People have done similar experiments now in rats, in pigs, and in humans with very similar results. And this seems to be a transport system which is conserved across many species. And it makes sense that there would be two distinct transport systems in the brain, because there are two distinct fluid transport systems in the rest of the body, as you probably all well know. And Jimmy Ward has done much of a career of work on the lymph system, which supplements, of course, that might even be too weak a credit for the lymph system. It operates in parallel to the circulatory system for blood and picks up water-like fluid, pumps it back into the heart after it trickles through the tissue, plays an important role in immune response, does all kinds of great things. And but there are no lymph vessels in brain tissue proper. And so it had long been a mystery because your brain is 2% of your mass, but takes 25% of your glucose. It doesn't make any sense for the brain to lack this second fluid transport system, sweep away waste and such. What we realize is there is such a system, but it looks a little different. It looks a little different. Okay, and it has important implications for human health. One is that it can sweep away normal metabolic waste whose accumulation correlates with diseases like Parkinson's and Alzheimer's. Another is that in pathological cases like stroke or cardiac arrest, this fluid transport system can malfunction and cause terrible swelling, which damages tissue. I'll tell you a little bit about that soon. So I'm going to tell a few stories about this transport system. I need to give you one more slide worth of background first. So it's analogous to the lymph system, but there are no lymph vessels in brain tissue. They do come to the edge of the skull. People only discovered that a few years ago. Instead, the path as we understand it is that fluid flows into the brain along perivascular spaces, surrounding arteries, somehow trickles through brain tissue. Some people think there may be perivascular spaces continuous all the way down to the capillary level and back out along the veins. I think the more likely explanation is that capillary spaces, perivascular spaces don't make it all the way down to the capillaries, and this fluid escapes and percolates through extracellular spaces before being taken back up by perivascular spaces around veins and nerves, and then along those making its way out of the brain and then to the spine and or lymph vessels that evacuate the skull. That's sort of the big picture idea. We're pretty sure about the influx routes because we can see them very easily. The efflux routes are a topic of ongoing study. We're less sure about those, but there has to be some kind of closed loop. And this big idea has been floated a few times in modern form. It goes back 10 years and was floated by Mike Nedergaard and collaborators, and they called it the glymphatic model because it's like the lymph system, but glial cells play a key role because the outsides of the perivascular spaces are formed from the end feet of astrocyte cells, and the aquaporin 4, which is expressed preferentially there, plays a key role. If you knock it out, the system doesn't work as well. I'm not going to talk more about that today. That's the reason for the G in the name. Okay, and that's the pathway I was sort of talking about. Yeah, and this 1985 Reynolds paper is an example of one that foresaw very much of the modern conception, but didn't have access to photon microscopy and some of the other modern instrumentation methods that have allowed us to look a lot closer at these things. Also, I need to tell you, these flows occur almost exclusively during sleep. There's little or no circulation through brain tissue when animals are awake. It may be part of the reason we sleep, we don't know. I'm going to show you lots of in vivo data, and I guess I don't want to take too much time, so let me just say two sorts. I've already showed both. One is with what we call transcranial imaging, relatively low magnification microscope, looking at a whole mouse skull, and the other is true photon microscopy through a cranial window. I'd be happy to talk in more detail, well, a little more detail. I don't do the experiments myself. You may well know this stuff better than I do. But the data is beautiful, and we like working with it. Here's another example of some tracer particles observed with 2-photon microscopy in a perivascular space surrounding a middle cerebral artery. And when we do this automated particle tracking, what we get out is a position and a velocity for each particle in each frame of the movie, which is quite a bit of data, actually. And we can use that data to draw pretty comet tails. we can use that data lots of other ways as well. For example, I could just average it all over time to show you what the mean flow looks like. Here's that. Speed is shown in color. You can see that flow is fastest in constrictions, which makes sense because conservation of mass, right? If you want to get more fluid through less space in the same amount of time, it's got to go faster. You can see the flow is really slow in the bifurcation of this artery, and that's biologically interesting because wastes are known to accumulate in bifurcations. You can see that the typical speed is on the order of 15, 20 microns per second. And that's interesting if you start thinking about fluid dynamics. We can calculate a Reynolds number, which probably you know is a dimensionless parameter that, among other things, predicts the likelihood of turbulent flows. It's speed times size divided by viscosity, and it's on the order of 10 to the minus 3 in these flows, which means laminar. Laminar all the way. We don't expect turbulence. We don't expect vortices. You can do simulations and neglect the nonlinear term, and Navier-Stokes is Stokes. So that's good to know. I said those things already. Yeah, okay. Another thing you can see if you look at this map of the speed is that it tends to be fast in the middle of the perivascular space and slow on the edges and very smoothly in between, which is just what you would expect for a laminar flow in an open space. And we were a little surprised by that when we first saw these because we wondered if these spaces might be better modeled as porous media, right? If they might be full of disordered tissue. But for these peel surface perivascular spaces, they're really not. And we look more carefully at velocity profiles over many experiments and found that when we constructed profiles along cross sections, like the one I'm plotting here in white, check that. The one I'm plotting here in yellow, which comes from in vivo data, they fit much better to a smooth, nearly parabolic profile of the sort you get for laminar flow, which we calculated using a best fit estimate of the cross-sectional shape. That's the thing in white. They fit that much better than they fit Darcy flow, which you might call plug flow, which would be a flat velocity profile, which is what you expect in a porous medium. That's shown here in gray. We did some other analysis too, but okay, from looking at how the fluids flow, we can tell that the surface perivascular spaces are actually wide open. Branching off from those are the penetrating perivascular spaces that dive into brain tissue. Those we don't know yet. And I guess Joe and I were talking just a few minutes ago. We'd love to know how porous those things are. That's in our modeling. I'm not going to show this stuff, but we put together a big brain-wide model, and we did sensitivity analysis to figure out which parameters most need to be measured to make better estimates. That's number one. So I'll just plant that in your heads. It'd be a really interesting thing for somebody to measure. Okay, another thing you can see from staring at these movies is that fairly few particles pass above or below this middle cerebral artery. And it's not because they're going out of focus and we can't see them. Once in a while we do see some go, but there just aren't many. And that actually makes sense when you look at the cross-sectional shapes of these spaces. They are not concentric circles around these arteries. They're sort of squashed and elongated. Here are two in vivo images and an artist's sketch. And my colleague Jack Thomas, a couple of years ago, got really curious about why these spaces might take on such funny shapes. And actually, he came to one of our biweekly meetings and he said, I woke up at 4 o'clock this morning and I had an idea and I just couldn't sleep. And here's my idea. He went to the chalkboard and he started writing. And he basically laid out the whole plan for a paper, which I think was the quickest I've ever written. And that's what I'll talk about for a sec. So, okay, credit to Jack Thomas and to Jeff Tiedoff, who was first author on this work. Jack proposed a simple geometric model for these spaces in which the artery is a circle with some radius R1 and the perivascular space is an ellipse with semi-minor and semi-major radii R2 and R3, R3 and R2, whatever. And we allowed for eccentricity too. So with 5 numbers, we can make a whole family of shapes. And if you then imagine fluid flow down a vessel with such a shape, well, it's covered by the Navier-Stokes equation. It's the momentum equation. Here it is. Rho's density, T's time, U's velocity, P's pressure, U's viscosity. It's famously hard to solve. But I told you already these flows are laminar, so we can pretty well throw away the nonlinear term. And we made a further dramatic simplification by considering only steady flow for starters. And doing all those things simplifies the equation to this Poisson equation I've written below. That one's much easier to solve numerically. So we started working on that for our family of many shapes. And we can produce velocity profiles pretty quickly. Here are 4 for 4 different shapes in the family. Color indicates speed. And what you can see is if we consider just concentric circles, the flow is very slow everywhere. As we increase the elongation, you get fast regions in the middle until the elongation becomes really extreme, and then it's slow everywhere again. And that makes sense if you know laminar flow, because the velocity has to vary smoothly over space, and it's got to be 0 at the walls. No slip boundaries. So in shapes where no place is far from the wall, you can't have fast flow. But to say it differently, for this family of shapes, moderate elongation allows the fastest flow for the same pressure difference. Another way to say the same thing is that the hydraulic resistance is lowest for shapes of moderate elongation, where hydraulic resistance is just pressure drop divided by volume flow rate. So then we kept going, and we calculated hydraulic resistance for many, many more shapes. Here I'm showing you 3 different curves. The 3 different curves are for different artery sizes, and on the horizontal axis, I'm varying that elongation from fully circular at far left to very long and skinny at far right. And what we found was, in every case, there is some optimal elongation for which the flow is minimum. I mean, the resistance is minimum, flow is maximum. And then we went to the literature, and though there were very few cross-sectional images published at the time, all the ones we could find, when we fit a circle and an ellipse to the actual image, had elongation and hydraulic resistance quite close to that optimal value. So we thought that was pretty interesting. When we calculated velocity profiles, they also are quite similar to the optimal one. It's easy to wonder if these shapes of these spaces actually evolved to minimize the hydraulic resistance and allow the body to pump fluid through these spaces as easily as possible. And that we published in 2019. Next, I want to tell you about a story about what can happen when things go wrong. So a stroke means blood is blocked to some part of the brain. And textbooks tell you about what happens when that occurs. Neurons starved of energy and oxygen are unable to maintain the ion gradients across their membranes. And so they dump their ions into solution, and then their neighbors are overwhelmed and dump their ions. And so you get this spreading wave It's a reaction diffusion wave. It's called spreading depolarization in the medical literature of ions being dumped into solution that crosses, perhaps the whole brain or much of it. And after the wave, you get edema. You get severe swelling caused by fluid rushing into cells. And that really damages brain tissue badly. And often, makes the difference. The amount of swelling often makes the difference between how much you can recover from a stroke or not. The textbooks also say that that fluid rushing in comes from blood. We wondered, given that there's another fluid transport system that people haven't thought about much, whether it might play a role as well. And to study that, Ting Du developed a protocol to induce stroke in mice where she injects a bead to block an artery. And she used genetically modified mice, GCAMP7, such that calcium signaling associated with neuronal activity causes fluorescence. You can just watch. So I'm going to play you a movie, and the top panel is neural activity observed that way. And the bottom panel is some tracer dye injected in the same way into cerebrospinal fluid. And you can see some pale green flashing, or maybe you can't even see it yet in the top video. It's just normal neural activity, it's not that important. You can start to see now in the bottom video some dye creeping in. That's normal transport of tracer. And there's the wave that you can see. And with it comes a huge inrush of cerebrospinal fluid. Let me play that again. There's the wave, and right behind it, a lot of tracer coming in the CSF. By the way, as you can see, the wave covers just one hemisphere of the brain, and the tremendous influx of cerebrospinal fluid occurs just in the one hemisphere of the brain as well. So we looked closer at that. We used a front tracking algorithm that my team had developed for totally unrelated stuff to quantify the local speed of the curves that separate bright green from not, or bright yellow-red from not. And by doing that, we're able to show that after the wave passes, fluid flow accelerates. So it does follow, even not just globally, but locally throughout. That made us wonder, what might the mechanism be? How is it that the spreading depolarization wave could be pulling in all this cerebrospinal fluid? And so we looked closer at arteries and perivascular spaces. Here's one-dimensional imaging of one artery and perivascular space over time on the vertical axis, space on the horizontal. And what you can see is that when the spreading depolarization reaches this particular artery at about 4 minutes, the neurons around start firing madly. You get a lot of calcium activity. That's shown in purples and yellows here. Sorry, I changed the color on you. And very soon thereafter, the artery constricts quite violently, leaving behind a perivascular space which is much enlarged and therefore fills with much more cerebrospinal fluid than what was there before. So it sucks in, the artery constricts when the wave passes and sucks in CSF. We can see that in movies as well. There's the constriction. Here's a huge inflow. Now this section of artery has grown back to its original size, but the wave has propagated onward. Sections downstream are shrinking and leaving bigger perivascular spaces. And so the inflow of fluid continues, until the wave finishes propagating. We were able to quantify that and compare to diameter. In this project, I think there were more than 20 co-authors on the paper. So people did radio tagging to show that, yes, indeed, the fluid really was going into cells and causing edema. It was not coming from blood. there was a lot of very careful checking by an amazing array of people. And we came to the conclusion that the early swelling, not all the swelling during stroke, but the early swelling from the first half hour or so occurs before the blood-brain barrier opens. And it's not due to fluid from blood. It's due to cerebrospinal fluid. And that has clinical implications because you might, if you're quick, give stroke victims drugs that hinder this sort of fluid transport to reduce edema. And that we published in 2020. We undertook a similar study thinking about cardiac arrest and found much of the same story actually and published that in 2021. Next, I want to tell you a story about some ongoing work. These fluids, after making their way out of brain tissue, have to make their way out of the skull if they're going to be cleaned and remediated and so forth. And the efflux routes are not so well known. It seems that a good bit goes out through the spinal cord. We also started looking at lymph vessels, and in particular cervical lymph vessels. And Ting Du did these surgeries as well. Credit her. And Adi Raghunandan is the postdoc who works with me, who's been doing the engineering bits on this. So they've been looking at cervical lymph vessels in mice. They do the same sort of injections, and what they find is that these tracer particles that we inject at the back of the skull, at least some of them, are eventually removed from the skull along superficial cervical lymph vessels in the neck. And we can track those and see their motion. And so we've been starting to think about how these flows work as well. We looked at mice of different age because, well, these diseases like Alzheimer's and Parkinson's come on with age, of course. And the exact mechanisms are not well known. Lots is known about Alzheimer's, of course, but I'm sure you've read, right? Alzheimer's drugs have been failing for the last 10 or 20 years, and there's kind of a crisis in the field because not enough of the precise mechanisms are known. We wondered if perhaps poorer efflux of these fluids from the brain with age might have to do with reduced function of these cervical lymph vessels. So we looked at mice of different ages, and we did find that the mean clearance speed drops with age. Here's some of the data. Raw data is in red, and the stuff in black is just a running average. We found it to be statistically significant. And about a 50% reduction. We also found that the contraction frequency of these vessels dropped with age. And that is also statistically significant and quite a drop, actually. And if you look at the typical contraction waveform, it changes quite a lot with age. So to make this plot, we took many, many measurements of vessel diameter, automated, right, over many cycles and phase average, right? So we called phase 0 the moment when each lymph vessel contraction occurred, and the intervening times we divided up into some number of intervals and averaged together, and everything was the same interval over many cycles. And so here's what it looks like, and the waveform's quite different. So this is, vessel dilation is normalized, where 0 means the minimum diameter and 1 means the maximum. And as the mice get older, it takes longer and longer for the vessels to dilate and reach their final maximum diameter. It's slower and slower. So we wondered why that might be, and got to thinking about tissue properties. We stained for collagen, we stained for smooth muscle actin. We found that the Collagen, though it gets a little dimmer with age, it's actually not statistically significantly different, whereas the smooth muscle actin really is. And so we wonder if the smooth muscle cells are just atrophying with age. And with that idea in mind, we tried to restore them. And Ting chose to use prostaglandin 2 alpha. It's a drug known to promote smooth muscle activity. It's given to induce labor in humans, for example. And she just put a couple of drops right on the exposed lymph vessel during the surgery while she was observing, and found that clearance speed goes back up a lot, even in very old animals, and that the vessel pulsations get stronger, even in very old animals. And so maybe something promoting smooth muscle activity could be used to offset the weakening effects of aging on flows like these and promote evacuation of waste. We don't know, but we're thinking about it. All right, how am I doing on time?
You have
a few more minutes. Okay, I'm going to tell. Oh, gee. Okay. I'm going to tell a couple more quick stories. This one is more work in progress, but I'm really excited about it, so I want to tell you about it. Particle tracking, we've done a lot of it, and we think it's pretty great. You get a lot of data. It's quantitative. However, that's not perfect. I can always dream for better because it's spatially sparse, right? We only get measurements where particles appear, and we inject as many as we can, but there could be more. It would be nice to have more measurements. They're sparse. And they're sparse enough that calculating accurate velocity gradients, right, if you want to know about strain rate, for example, we really just can't do it. It's too noisy. Also, this is 2 velocity components measured in 1 plane for a 3-dimensional flow. We'd like to have 3D information. I showed the Navier-Stokes equation already. Pressure, of course, plays a key role. Just measuring velocity, we know next to nothing about pressure. We'd really like to know about pressure, especially because a story I didn't have time to tell today is all about what are the pumping mechanisms driving these flows. And the short answer is nobody's sure yet. A longer answer is it certainly has to do with expansion and constriction of arteries. And functional hyperemia can do a lot of it. And well, there's some other things going on. But if we had access to the pressure, we might be able to learn a lot more about what exact pumping mechanisms are at play and their relative importance. We'd like to have access to wall shear stress because it's such an important signal in so many biological situations. We suspect it might be important here too. And we can't get any of that out of direct particle tracking. But we started a new collaboration with George Karniadakis at Brown, and we're starting to squeeze some of this extra information out of the data. And we're using a technique called artificial intelligence velocimetry. The people doing the heavy lifting on this are my postdoc, Kim Boster, and Shengja Kai, who was a postdoc with George and is now starting his own lab. And this is kind of a busy slide, but here's the idea. Neural nets, they're good at making up answers for things, right? And we don't really understand how they make up an answer, but they'll make up an answer. And one common thing to do is to give them a lot of training data and to tell them to try over and over and over again to make up answers that match the training data. That's something. I never liked that idea for fluid dynamics because we know the physics. We can write down Navier-Stokes. Why would I ignore that? And George had this brilliant idea to go further. And so what we do in artificial intelligence plosimetry, we have a neural net, we ask it to make up a velocity field and a pressure field. And we ask it to minimize error, but the error has three parts. One is, minimize error with our particle tracking measurements. Try to match the measurements. The second part is, minimize error with the no-slip boundary conditions. We tell the neural net where the edge in 3 dimensions of the perivascular space sits. And we know velocity should go to 0 there. And the third bit is try to match the fluid mechanics. So we can write down the Navier Stokes with the momentum equation and the mass equation. And if you put all the terms on one side, it's 0 equals whatever. And with the solution the neural net made up, you can plug it in, and if you don't get 0, it's wrong, at least a little bit. So we ask the neural nets to simultaneously match the data, match the boundary conditions, and match the physics. And we feed in particle tracking data. We do phase merging, so it's not quite so sparse. We feed in 3-dimensional shapes of the perivascular spaces in which we measure these flows, extracted by segmenting Z-stacks from the 2-photon microscope. And we can infer velocity, for example. We can do it at high spatial and temporal resolution. And to check, so first of all, when you do things this way, the fact that the neural nets are a black box and you have no idea how they make up their solutions, to me, it's a lot less troubling when I can look and see how close did they get to matching the real physics that I trust. Beyond that, we also check to see how close they get to the original data. So we trained on 70% of our particle tracks, and then we compare the results to the remaining 30% of our particle tracks. That's what I'm showing you at right. I've got 2 different single number measures of speed, the root mean square velocity and the downstream velocity, plotted as they vary over a single cardiac cycle. I should have said that. These flows pulse, and they pulse in lockstep synchrony with the cardiac cycle. So that's the phase averaging we're doing here. We get a close match. Seems to work pretty well. And we can ask for velocities anywhere in the 3-dimensional volume, even though we've only trained on 2-dimensional data. We can also ask for pressure. So here's one slice of the 3-dimensional pressure field as it varies throughout a cardiac cycle. So here, little t is time, and big T is the duration of a cardiac cycle. In a mouse, an anesthetized mouse, the cardiac rate is about 5 hertz. So we can get pressure out, which we're really excited about, and we're sort of just getting to work with this. From 4 mice, we get about 2 millimeters of mercury per meter as the average pressure gradient. That's actually pretty close to estimates from a couple of recent first principle simulations, right, where they weren't using experimental data. And I think one used a domain shape that was taken from in vivo imaging, and one made up a domain shape that was reasonable. But we're pretty close to the simulations, so that's encouraging. One implication right away of just the magnitude of this number is it's small enough, it's pretty unlikely to substantially deform the surrounding brain tissue. And that's important because it changes the flow a lot if you're losing energy deforming the tissue with every pulsation. As I said, we hope to use this soon to gain insight into what mechanisms might actually be driving these flows. And okay, I'm gonna tell one more quick story because I had chats with a couple of people about this today. We've been thinking a little bit about drug delivery. The blood-brain barrier prevents many molecules from crossing from the blood flow into the brain at appreciable amounts. So it's hard to get drugs to brain tissue. And one idea is, since there's a fluid transport system circulating water-like fluids around and through brain tissue, put some drugs in there, use it to deliver them. We're not the first to have that idea, but we did some work on it. And an idea that is novel, is not mine, it was Migan's, but it's a great one, is facilitate the flow by expanding the spaces between brain cells by injecting hyperosmotic solutions, right? So I said earlier, these flows go almost purely during sleep. It's also known that the fraction of brain tissue which is in between cells and open, where such flows could occur, trickling through the tissue, is like more than 20% during sleep and 14–15% during wakefulness. So the spaces get a lot bigger to allow flow to happen during sleep. The idea here was use osmosis to enlarge the interstitial spaces in the same way to allow better drug delivery. And we didn't actually inject drugs. We injected tracer dye, but we were able to see nonetheless that over time with this osmotic manipulation, we had much more tracer entering deep into brain tissue than without. And we can quantify that. It works pretty well, and we published that a few years ago. Okay, with that, I'm sure I should wrap up. I want to thank my research team and make it fun to come to work every day, and did most of the heavy lifting for much of what you saw today. And we have openings for PhD students, postdocs, and a data scientist. If you're looking for research to do, I'd love to talk to you. Thanks for your time.
Thanks so much, Doug. It's always a pleasure to see your work. Maybe I'll have a question to start off. I think, I'm really interested in this idea of it only happens during sleep where you have this experience, right? I'm sure you get this question all the time. I mean, I guess when you look at your models for disease, do you do those on live animals, awake animals, or sleeping animals? Do you see a difference in really sort of the frontal wave you saw earlier? Do you see a difference between the waking or?
Let's see. I guess there's much to be said. So it's well known that poor sleep correlates in humans with these diseases, right? Bad sleep definitely correlates with Alzheimer's. We also know that this stuff happens much more in sleeping animals than awake animals. My colleague Lauren Hablitz did a really nice study where she tried a bunch of different anesthesia, and some of them are much more sleep-like than others. So you have to be careful when you do the experiments if you want to see any flow. Let's see. You were asking about earlier
on, you were showing some models of disease in mice, I believe, like a stroke model.
The stroke model.
And
okay.
Are they awake or sleeping? Would there be a difference there?
Yeah, fair question. That was entirely done under ketamine xylazine anesthesia, which is the one that we know of that makes flows that are most sleep-like. So we haven't varied sleep-wake state and tried that. That's an interesting question.
I guess the other question I'm sure he gets all the time is, do we know what controls the flows what stops the flow during wakefulness versus?
We don't know for sure.
Yeah.
We don't know for sure. To me, my best guess is this change in the porosity and the size of the interstitial space, where it gets much smaller during wakefulness. It may single-handedly be enough, because hydraulic resistance goes as width to the 4th power. So if you change the width of things a little bit, you get a huge change in hydraulic resistance. It's pretty clear now and pretty well documented that flow continues during wakefulness, but doesn't go through the deep brain. So when people look at awake animals, or when they look at animals who've been given anesthesia that produce a more wake-like flow, you still get a lot of fluid motion around the brain, but it seems to not go through. It seems to get shunted in the subarachnoid space, like straight out the spine, straight out the lymph vessels.
Yeah.
So my best guess is the pump is always on, and the sizing of the interstitial spaces in the deep brain is the main valve mechanism that controls whether stuff goes through or just around. But I'm not totally sure what the pump is, and there may be a lot of refinement to do about that.
Big questions. I'm also reminded that when astronauts go to space, that their faces swell up because gravity just drains their limbs right downwards. I'm just wondering if sort of maybe lying down is another contributor to sort of opening something up. And
could be. However, mice don't change their posture much when they sleep.
There you go. Yeah. Fair enough. Other questions? Yeah, I'm sure you have plenty.
Yeah, thank you so much for this event. It's very interesting to realize that these fluid activities kind of heightened when during sleep, and it actually makes this area kind of open and explored because there's a lot of diversification can get there. I'm just wondering, have you tried to, are you guys thinking along the line of this motion affecting sleep? It looks like sleep pattern affects the motion, but does it determine sleep pattern at all? Or as this also affects the brain activities and hence some of these pathological cases or situations?
So your question is, does the flow determine sleep state or influence it? Is the causal coupling the other way or maybe both ways? That's an interesting question. Have we done anything like that? I don't think so. That's probably a wide open question. People manipulate sleep states to see how the flow changes.
What's investigating? Because we're actually at the moment looking at it from one angle, as though sleep determines what is happening. But could it also be that this flow pattern determines sleep pattern that now affects humans in pathological cases or pathological situations?
It's possible. That's possible. Most of the work that's been done so far on this field is either modeling, from numerical or pen and pencil modeling, or in vivo. There's very little in vitro going on, and there's very little manipulation of the system with anything other than just drugs. And this might be the kind of question where an in vitro approach would be the right one, and there's opportunity for that.
I'm fascinated by this idea that you get a sudden drop in cerebrospinal pressure during stroke. Have you basically invented a monitoring technique for old people? Like, we can set up an alarm when you see a sudden drop in pressure, and the ambulance is on its way before they've even hit the floor.
That's interesting. I hadn't thought about it. I don't know how you'd measure it.
Well, you mentioned that there's a pocket of cerebrospinal fluid at the back of the head. So you could
Maybe you could get it there. Maybe you could get it there. That's a really interesting idea. I don't know.
Jimmy.
So you've been able to measure velocity, and maybe you can do this with the cardiodocus method. Can you get volume flow rate?
We can do, we're getting better and better at it.
You either need some assumptions or a good 3D reconstruction, right?
Right.
So, I'm just curious, because I think that would be useful to get a volume flow rate number and then take some good images of mouse blood circulatory systems and try and estimate what the total outflow of this system is. And I'm guessing it, I mean, you're based on a velocity of, what was it, 20 microns per second. I mean, that's low, right? So it's going to be something that could easily be taken up entirely by the lymphatics, not very many lymphatics draining it. So you know, there's so many mysteries around this system. At least some sort of conservation of mass or magnitude analysis might be quite useful.
So we have done some calculations of those sort. We can get volume flow rate really well with the AI stuff.
Yeah.
I forget the numbers off the top of my head. One thing we found when we estimated system-wide volume flow rate, basically by using the 20 microns a second, using our best fit elongated cross-sectional shape, and multiplying by 5 because the middle cerebral artery is sort of one of 5 big ones in the brain for the mouse. We got a number, and it was something like 3 times the textbook number for production rate of cerebrospinal fluid in the choroid plexus of the mouse. So one conclusion could be it's not just production in the choroid plexus pushing everything, and there's actually got to be some recirculation. Another conclusion could be measurements of production in the choroid plexus are not all that reliable. They're kind of broad in the literature.
3x in things like this means they're equal.
Yeah, okay.
Right? So,
Yeah, okay.
the other thing so going back to what Chris had just asked about, when you show that vasoconstriction after stroke, so was that stroke model ischemic or hemorrhagic?
Ischemic. I should say that.
So you've reduced the pressure in that artery, and is that why it's constricting, or is it an active constriction?
The constriction follows the spreading depolarization. So like you get the pressure drop as soon as the bead hits, but 8 minutes later the constriction starts.
Yeah, sometime later. Okay. Yeah, it's almost certainly active constriction.
I think so. I think the ions are driving it.
And if nobody else has a question, I have one more, but we can give other people questions.
I just had a quick question just about the drug treatment with lymphatic function. Was that a transient effect or was that permanently restored?
That's transient. It's transient. Yeah,
How
yeah.
long does that last?
Oh, that I don't know. Our experiments are on the order of half an hour. It lasts that long.
Okay.
Whether it would,
Return to baseline? Like that significant decrease or?
I don't think we tried. I don't think we've watched long enough. We run out of tracer particles, after half an hour, and then we can't measure flows anymore. Silly technicality, but I don't think we've tried. That's an interesting question.
It'd be really interesting to know just how much that transient lasts for.
Yes.
Thank you.
There is one question online from Kim Parker, I think. In part 1 of your talk, do you observe pulsations of the larger arteries during the cardiac cycle?
Do we observe pulsations? Yes. So that's one of the stories I didn't tell. We do see pulsations of the arteries during cardiac cycle. We do see that, you could probably tell that these flows are pulsatile. I guess I said it once already, but we can quantify that. In this part of the brain, they pulse in lockstep synchrony with the cardiac cycle. People have reported cerebrospinal fluid flows following respiratory cycle elsewhere. Maybe they do, but here it's totally cardiac. We have also seen that the fluctuations of flow speed match in magnitude and waveform shape quite closely to the velocity of the adjacent artery wall, which is some pretty strong circumstantial evidence that artery pulsation has a lot to do with driving this flow. We also manipulated artery wall motion by raising the blood pressure to stiffen the arteries with angiotensin II. We found that the blood pressure did rise, we found that the artery motion did change, and we found that it did reduce cerebrospinal fluid flow. So this is another set of facts that suggest artery motion has a lot to do with the pumping. Caveats are, if you try to reproduce, people have tried to reproduce that in simulations and it hasn't come out well. So that's a whole other story, but there's a lot to the artery motion and we look at it closely.
I mean, as the arteries get smaller, the velocity is increasing in the arteries as well, and the resistance in the perivascular space is going down, right? So it should all sort of go together.
Right.
Jim, you have another question? You want to?
I was just, if you can go to the next slide, or maybe the one after it. So yeah, so my recollection was that the perivascular space around, if you that figure in the upper right corner.
Yes.
Right, so you're showing the perivascular space in light blue around the artery, same thing around the vein. My understanding is they connect in the subarachnoid space. Is that not the case?
You know, the jury is out on the anatomy of the subarachnoid space. The simplest explanation is yes, but then you start asking about what sets the flow direction.
Yeah.
And also you have to start wondering about, is the brain really just, does the brain not separate the clean fluid from the dirty fluid, if this is supposed to be a waste evacuation mechanism? And those at the moment are open questions. When I started this, I thought surely that anatomy was known, but it seems people still debate about it.
Right, thanks.
On the corner back there.
It was observed that prostaglandins, they affected the smooth muscle contraction and improved it in a way. And I think that would help reduce the stroke risk because it would prevent accumulation of the fluid.
Yes, it might. Yes, it might.
Maybe that would be used as a clinical intervention in the short term or the long term, because with aging, as you mentioned, the smooth muscle cells deteriorate and do not contract as well. So maybe it could be a long-term intervention as well as short-term post-stroke.
Maybe so. Yeah, just to repeat for the people on Zoom, the comment was maybe you could give prostaglandin to promote the skull evacuation by the lymph system in the case of a stroke. Yeah, that might work. I don't know.
Maybe we can find a way to help it get into the cerebrospinal fluid.
Mm-hmm. Mm-hmm.
Great, plenty of great questions. We do have a social after this. I think there's drinks. Is Kemi in here? No, I think Kemi had stepped out. There should be drinks somewhere. I'll figure out where they are. Okay, thanks, Chris. 324. So if you have other questions, please come and chat with Doug and have a drink with us. Why don't we thank Doug one more time for a great talk. Thank you so much.